Development of a microfluidic research device to investigate cancer metastasis. Metastasis is the process through which cancer cells from a primary tumor enter the bloodstream and form secondary tumors at distant sites. This is how cancer spreads. Metastasis is a major predictor of morbidity and mortality in cancer patients. This process begins with an oncogenic transformation event, followed by angiogenesis. The tumor then invades the surrounding connective tissue and causes changes in the extracellular matrix and adaptation of the tumor microenvironment. Cells intravasate or enter the bloodstream and become circulating tumor cells. Most of these are rapidly destroyed by the immune system, but surviving cells can arrest in the capillary beds of organs or other tissues. They then extravasate or enter the organ, and a metastatic site is established as tumor cells grow and proliferate. But the details of this process are poorly understood. So we devised a method to study metastasis that attempts to combine the physiologically relevant data we can generate with in vivo models with the high throughput, rapid, low-cost nature of many in vitro methods. Our experimental platform is a microfluidic device composed of three main parts. The first is a valveless pump that reproduces peristaltic flow throughout the system that will provide nutrient and oxygen-rich media, kind of like the heart. The second are tissue channel interfaces, one well for cancerous tissue and one for healthy tissue that might serve as a secondary tumor site. These features alone allow us to conduct studies involving transformation at the primary tumor site and the process of forming a secondary tumor. But circulating tumor cells are a valuable type of cell to study because the understanding of genetic changes that allow for circulating tumor cells to enter the circulation and spread cancer to a secondary site are great targets for novel drugs and therapeutics. With this in mind, we've included a dielectrophoresis sorter to allow circulating tumor cells to be separated from the primary blood flow without the use of antibodies or other methods that may impede our ability to do downstream analysis. Together, the system allows us to study metastasis, circulating tumor cells, and even potentially to do high-throughput drug screening. For the purposes of this project, we chose to focus on a specific experiment. We seed one experimental chamber with NDA-MB231 breast cancer cells, an aggressive triple-negative form of breast cancer, and the other chamber contains healthy brain tissue, separated from the circulation by a simulated blood-brain barrier. This serves as a potential metastatic site.